So, yesterday we did not complete our discussion on the RNA based differential regulation, right. So, what did we discuss at length yesterday? What is the major theme? Splicing, alternative splicing and its impact, right. And then we discussed about how microRNA also contribute to regulation. So, I forgot to recollect that. So, remember I told you some of the mechanisms by which regulation happened, we did not know till 2 decades ago. So, that was I was referring to microRNA. Okay. So, there will be microRNA that will be encoded by the genome and that will regulate the translation, was not predicted, and only through genetic mutant screens when people found, we discovered that, and then we realized it is there in all organisms and its effect is lot more widespread and general to the regulation than what originally was thought. Okay. So, that is why people called the non-protein coding regions as junk DNA, right. So, now we know probably there isn't a base there in the genome without a reason. So, today what we are going to do is we are going to look at localization also plays a role, okay, where an mRNA is present and when is it present also matters. So, here is one good example coming from Drosophila oocytes, okay. So, during the oocyte development itself, this is even before fertilization, there is an asymmetric localization of molecules, okay. A good example is this nanos mRNA, okay. So, once it is deposited in the oocytes by the nurse cells as the oocyte grows, the nanos mRNA molecules move towards the posterior primarily by diffusion, but then at the posterior they are bound by a protein complex. They are primarily another RNA binding protein called Oscar binds and protects the nanos RNA from degradation, and also Oscar activates nanos translation. So, here what you are seeing is RNA being localized within a cell itself to a particular area, in this case posterior, and there only it gets translated. So, the unlocalized nanos RNA is not translationally active, okay. Diffusion and local anchoring. And another example we are going to see is again from Drosophila itself, this HSP83 mRNA that is protected from degradation only when it is in the posterior. In other places a deadenylase complex is going to bind and that is going to degrade the RNA, okay, localized protection. A third aspect of this is transport of mRNA, okay. So, again these are all from the Drosophila embryo itself. So, in oocytes we are looking at. So, Oscar mRNA in these microtubules by the motor proteins get transported to the posterior. At the same time, on microtubules pointing towards the opposite direction, motor proteins transport bicoid mRNA to the anterior. So, this is how asymmetric localization happens in the very early embryo itself. In this case it is oocyte, it is not even fertilized. And so, therefore, when you keep following like yesterday's question, the cascade, transcription cascade, this one is specific to this tissue, then what made it specific to this? Like if you keep going back and back, you will all the way go back to an one cell embryo, okay, to the zygote. So, therefore, the differential gene expression starts in the one cell embryo by asymmetric localization of molecules in a single cell. So, cytoplasmic symmetry is converted into an asymmetric cytoplasm, meaning different parts of the cytoplasm has different components. So, as a result, people think this is the starting point and therefore, embryonic polarity is the most exciting thing, like that some people think and they focus. Okay. So, there are a lot of labs that focus on how polarity is established in the early embryo in different model systems. Okay. So, this is for now is our brief overview of how differential gene expression is brought about. Okay. First we learnt that it is primarily the nuclear material that seems to be controlling development. Okay, the early genetic evidences told that, and then other experiments like for example somatic cloning experiments told there is a genomic equivalence. This convinced us that therefore development must be brought about by differential regulation of gene expression. So now we saw how that might be accomplished. Okay, so various aspects starting from chromatin structure to mRNA asymmetric localization, right. So now we are going to take a huge detour. Okay, we are going to move from developmental biology to genetics, because I feel maybe this class is different because some of you have already learned a lot of genetics, but usually what I find in a developmental biology class, at least here, like meaning in India, most students have not learnt even the ABC of genetics, and they come and sit in a developmental biology class, and the whole of development has been learnt only through the methods of genetics. So I used to find it hard to teach developmental biology when students did not know the fundamentals of genetics. So I usually have a few lectures fully dedicated to genetics as part of this course. So this course actually should be aptly named as Introduction to Developmental Genetics. Okay, so all of you know Gregor Mendel was an Austrian monk, but there are some little bit of history that is worth recollecting, at least in the Indian context, where becoming high school teacher is not like a high flying career goal for most youngsters. Is that a fair statement? Right. Mendel seriously tried to become a teacher and couldn't pass the exams and could never become a teacher. Okay. So in many of the countries where education is taken very seriously, becoming a school teacher is a career goal, and it is not an easy thing to become a teacher. If you teach, you will quickly realize that. It takes a lot of clarity in your understanding of the subject from multiple angles of viewing those concepts, and great communication ability. Communication ability does not mean flawless, grammatically perfect language. You need to know what to say at what point. The spatial temporal regulation of expression becomes important. Okay. So he failed multiple attempts, he could not succeed. Then he realized, okay, monastery is probably the right place to spend the rest of the time. I think this will be useful for you, that is why I am saying this. So let us now move on to his experiments. So before moving on to his experiments, I would like to tell the historical context in which his experiments were done. So the historical context at that time was that people knew genetic information is transmitted from one generation to another generation. Okay, if you think genetic information itself is little technical jargon, let us say biological information. What is biological information? Only a Homo sapiens make another Homo sapiens, right? Only a black buck makes a black buck. So that already tells you that the black buck has the information to make another black buck. Only humans have the information to make another human being, right. So obviously there is information, and that is being transmitted from generation to generation. So the branch of science that tries to focus on how this information is transmitted and what is this information, are there general rules that govern this transmission process, that is genetics. Okay. So people knew this, transmission happens, information must exist, but people did not know how it happens. There were multiple theories, okay, including that the sperm head, that cartoon that is familiar to you. But the predominant prevailing theory at that time was called blending theory, meaning the information coming from maternal and paternal source are blended together to generate the next offspring. But then there were contradiction to this in everyday observation. Like for example, let us say you have straight hair, okay. Your father has straight hair, your mother has straight hair, but your paternal grandmother had curly hair and you have curly hair. How blending will explain that, right? So, that curly hairness has never been blended with straight hair and became an intermediately curly in your parents. They both have straight hair. So, there itself you have a problem with blending, but in the F2 generation, that is in your generation, you get your grandmother's curliness in your hair. Is this observed or no? Do we see that? It may not be curly hair itself, but there are many other features like this. So, you have certain features which resemble one of the grandparent as we said, but not there in your parents. That is one. And second, plant breeders quickly realized hybrid vigor happens in the hybrid, and then in F2 again you get the grandparental qualities. So, like this there were multiple evidences that contradicted blending theory. So, people were interested in finding out how exactly genetic information gets transmitted. So, that is the context in which we need to look at Mendel's experiments. So, let us look at his experimental strategy first. So, gourd and pea plants were readily cultivatable in that part of the world, and locally among the farmers he could find seeds for gourd and pea having different phenotypes. Let us use the word phenotype right away. Normally you do not use those technical jargons till the end of discussing Mendel's experiments, but for convenience we will use, because we are familiar already, genotype and phenotype. So, like for example, flower color variation here, purple and white, and similarly many other such features, like tall plants, short plants, flower coming at the terminus, like meaning at the top of the shoot, and flower axially, meaning on the side, like this. There were many variations that were readily available, and therefore he took them and cultivated and tried to do genetic crosses and observe what happens. So, this is the experimental strategy. Okay, so I am not sure whether this was discussed and described in detail in school. It was not done to me, and that is why I am assuming your schooling was much like mine, and therefore what I missed there I focus attention here. So, if it is repeat, just bear with me, it is only today. So, here the experiment you are doing is, first you need to consider, unlike us humans, many flowering plants have both gametogenesis. Same flower can make sperm and oocyte, okay, pollen grains or the ovum, okay, in the carpel, stigma and carpel. So, all this they can have, like for example, if you look at it, the stamens contains the pollen grains here, and this carpel is going to have the eggs there. So, it is there in the same flower. So, that is something you need to know. The many flowers are bisexual. So, now and then let us take a bisexual flower and look at what might be happening. Both the gametes are coming from the same genome, right? And if you have taken a gourd and pea variety that is giving purple flower for several generations in continuously, so then its genome definitely contains the information to make only purple color and probably not for any other color, maybe hidden, but we do not know. So, now if I want to cross a white one and purple one, and the flower color is what I want to follow to discover what kind of rules govern genetic transmission, then I can't allow the same flowers, both the gametes to fuse, right. So, that is something you have to address right away. The way it is addressed is, you open up the flower bud even before it is ready for pollination, etc. You cut the stamens out. So, therefore, the plant that you are selecting as the female source of female gamete, you are not going to have the chance of its own pollen fertilizing the ovules. So, that is cut, okay. And then it is also covered with a paper bag kind of thing, so that randomly pollen from the environment does not come and fertilize. Now, then you take a flower plant that you like the white color one in this experiment, in this particular example, then with a paintbrush you dab a little bit of the pollen grains from that, then open up the cover and go and spread it on, coat it on that carpel there. So, in this way you are controlling which pollen is going to fertilize the oocytes of the plant that you have chosen as the female. So, this is how the genetic cross is done, clear. So, now the fertilized one is going to grow. So, the ovary is going to form this pod, it makes more oocytes there, and therefore, it is going to make several seeds. So, each seed is an embryo, okay. It had one pollen grains genome and one X genome fusing together to make a diploid. It is just that it has it is like in some of the mammals, like if you take mouse or dog, you have litter mates, right, it is not like one only. So, like this you have multiple of them, then you sow those seeds and allow them to grow, and when they grow then you look for the phenotype. So, this is the experimental setup. When it grows, now if you are focusing on flower color, then you have to wait till the plant grows up to the flowering stage, and when it flowers then you see the color, and the flower color here is the phenotype, and whatever may be the genetic information that is contributing to it, that we call as genotype, okay. So, we are starting those terms early. Is this clear how this experiment is done? And therefore, when we are going through the experiment, you will be able to imagine the experimental methodology, then only you will be able to understand and properly explain the outcome. Now, when he actually did an experiment like this, so one thing he made sure is that when he collected the seeds, he ensured that that seed is pure breeding variety, okay. So, that is a technical term. What is pure breeding variety means? For several generations in a row, that phenotype only was displayed by that particular set of seeds, okay. Let us call it as a strain. So, that produced only that one variety, it was not giving rise to other phenotypes. So, that is what is pure breeding. So, now when he did an experiment like this, as you see in this cartoon, all the flowers were like one of the two parents, in this case purple color, okay. This was the experiment that he got at the first step, and he called this as the filial generation 1, and therefore, this is F1, where this is the first generation after this genetic cross or this cross mating, okay. So, now let us look at what happens when you go further. So, this is the parental generation or P generation, then filial generation 1, where all plants produced purple flowers. So, now what he does is, here he does not open up and cover it and selectively bring stamen from somewhere, he allows them to self pollinate among themselves. So, when he did that and then collected the seeds and sowed it, and when they grew and new flowers came, the F2 generation comes, there he ended up seeing roughly 3/4 of the plants produced flowers that were purple color, okay, the same color that was there in F1 generation, okay, not the color that disappeared in F1. The color that was not there, or the phenotype that was not there in F1, the phenotype of one of the two parents that was not there in F1, appeared in F2 in 1/4 of the plants, okay. So, this is all the experiment and the result. Now, shall we try interpreting it, what this means? because Mendel did it. Now we know whole lot of the rules already and we have studied this. So now looking at the experimental data and forgetting what we have learnt earlier in high schools, can we interpret the results now? So how would you interpret this? Because he had to interpret, and that is what we are memorizing in school. So this has the summary, okay, for one cross. So this is the parent, pure breeding parents. This was always purple, purple, purple for generations. This was always white for generations, white, white, white. Then in F1, the purple only is there, the other one is not there. And in F2, when you do selfing here, okay, so its own stamen and its own eggs, then you get 3/4 that purple color and 1/4 the white color. So based on this observation, can we interpret? So the experiment was not done just with the flower color alone, and it was done with axial bud versus terminal bud, yellow seed color, seed coat to green, and seed shape round or wrinkled, pod shape inflated or constricted, green or yellow pod, tall plant or short plant or dwarf plant. Like that he took multiple phenotypes, okay, one phenotype at a time, and did mating with the opposite or contrasting phenotype on the same thing, like color or height or shape, where you had a distinguishable another feature, okay, like purple one color, white another color, like that. So he did multiple experiments. In all of them, this is the kind of numbers he got. My teachers never said it is 705:224 and therefore it is 3.15:1, they said it is 3:1. So we never saw experimental data like this. So here you see the numbers here, these are the actual numbers that were counted. Is this telling you something? Like, okay, color reappears, one that was not there in F1 reappears in F2, killed the blending theory, okay, like final nail on the coffin for the blending theory. But anything else, fine, that theory is wrong, but are you coming with new theory? You have to interpret the observed results, right? So why in F1 1 did not show up? Why 3:4 ratio all the time? It is not random. Maybe purple and white you got 3:1, that is random, you can say, but why in every one of them 3:1? Right. So the first thing is you need to assume that probably I am getting information from both parents. Parent 1 gave the information to make purple color, parent 2 gives information to make the white color, okay. So then you need to invoke this dominance recessive to explain why one of them is absent in F1. And then to get to 3:1 ratio, that assumption that you have two sets of information is crucial, and only then you can work this out. So this is what Mendel worked it out. So he assumed that every organism, since it has two parents, sexually reproducing organism, two parents, it probably has two copies of the biological information or genetic information. And during gametogenesis, he assumed, okay, this is before meiosis, okay, cytogenetics is still not there, microscopes are still not being used extensively. So he assumed one copy of the information only goes to a gamete and not the other one, and therefore you use two letters to denote a genotype. This is a question I often ask in some students, okay, in PhD interviews. I will ask them, go and draw monohybrid cross. They will go and start. Their usual thing I find is not purple and white, it is usually tall and dwarf. They put capital T and small t. They will put TT. Then I will stop and ask, why two letters? You want to denote tallness, put capital one single T, I will understand, you know, you do not need to write twice. And there itself they go blank, they do not know why we use two letters. So the reason we use two letters is based on the assumption that there were two parents for that individual. So there were two copies of the biological information, and in gametogenesis each gamete gets only one copy. So if I am going to assume, so if I use capital P to denote purple color, and the gamete contributed by this therefore brings one copy of that. Now, this one similarly brings one copy only of the white color, which is denoted by small case p. Now, when these two fuse and make a diploid cell, that is going to be capital P small p. Now, if you use what she was pointing out, dominance and recessive, then let us say the purple is dominant, like purple is going to show up if it is present even in one copy, then this is purple. So, this kind of assumption and fusion of the gametes would allow you to consider this being all purple. So, that would explain that. Now, let us look at gametogenesis in the F1 generation. Half of the gametes will have capital P, half will have small p. Now, if there is random fusion of capital P and small p, then you will have this Punnett square representation. Okay. So, this makes only you got half of them capital P and then half of them will be small p, okay, in egg as well as in the sperm. 50% will be this and 50% will be small p. Similarly with the sperm. Now, if you have a random fusion what I mean is there is no bias here at both levels. Gametogenesis, there is no bias to having capital P or small p. So, therefore, 50% of the gametes have capital P, 50% of the gametes have small p. And similarly, when the two gametes fuse, again there is no bias. Capital P is not looking for a sperm with capital P, it fuses with either one of the two. Similarly, sperm does not have preference for either one of the two types of oocytes. So, when you have total randomness, then you will end up getting this combination giving you 3:1. So, this is what Mendel deduced from his experimental observation, and by considering that the parents chosen had 2 parents each. Okay. So, that is the thing, and based on this he proposed law of segregation, meaning the a particular genetic information, let us now use for convenience the word gene. Okay. So, probably exists in multiple variants. For example, one variant makes this purple color phenotype, another variant makes this white color phenotype. And these two variants, or how many ever variants exist, they do not influence each other when present together, as you see in F1, right. The white did not make purple to become something else, or purple did not influence the other, they stay together, during which one does not alter the other one, and they independently segregate when gametes are made. One does not influence the other one, and then there is a random fusion of the gametes. So, that is what is law of segregation. So, now okay, this is the time I should actually introduce these words, but you already know what is genotype and what is phenotype. The genetic information or the informational constitution is what you call the genotype, and phenotype is what is the visible biological outcome, okay, biological function, in this case the flower color. So, when you have the same type, both alleles are identical, both the variants. So, the variants we use the word alleles, okay. So, when both are identical, you call homozygous. This is homozygous, this is homozygous, and when they are not, it is heterozygous. Okay. So, these are the new words here, homozygous, heterozygous. Then second, you need to consider genotypic ratio can be different from phenotypic ratio. Here the genotypic ratio is 1:2:1, meaning 1 homozygous for the capital P, 2 heterozygous and 1 homozygous for the small p, 1:2:1, but phenotype ratio is 3:1. Okay. And this difference is explained by one of them being dominant over the other. So, now to address that, how do you find whether it is capital P or capital P small p, and that is where you do a test cross. So, test cross is when you have an organism displaying the dominant phenotype, and if you want to find out whether its genotype is homozygous for the dominant allele or it is heterozygous for the dominant allele. To address that, you do a genetic cross with parental phenotype, okay, where it is homozygous for the recessive phenotype. So, that is what is back cross, crossing the selected one with recessive phenotype, that is the parental recessive phenotype. So, since you are crossing back with the parent, many times you call it as back cross, but a good way of saying it would be a test cross, because it is a cross to test. And when you do that, what will happen, going by the logic that we have just understood, this will make only 2 types of gametes, while single type of gamete, while this will make 2 types of gametes, right. Now, if this were to be heterozygous, it will make 2 types as shown here. In this case, all of them will show the dominant phenotype, but on the other hand, if it is going to be let us say if it is going to be homozygous, it is going to make only one kind, and then when you take from this, it will be like your F1. But on the other hand, if this is having heterozygous genotype, it will make 2 kinds of gametes, and these 2 kinds of gametes, as you work out this Punnett square, you will realize half of them will end up giving you the recessive phenotype back, because this gives you only one type, and half of them, like this one with this or with this, will end up giving this. So, you will get a 1:1 ratio. If it is heterozygous, you will get a 1:1 ratio. If it is homozygous, you will only see the dominant. So, this is how you will determine the genotype, whether it is homozygous or heterozygous for the dominant allele. So, this is the back cross or test cross, right. So, you remember crossing with the homozygous recessive parental phenotype. So, the next experiment is now relatively easy to follow. So, in the previous one, it is only looking at one particular phenotype, okay, flower color alone, not 2 phenotypes at the same time. Now, looking at 2 of them, let us say, or multiples of them. To start with, let us say we are taking 2 of them. Will one influence the other? Meaning, let us say if I am a plant, tall plant with purple flower, okay, and the other one is a short plant with white flower. Now, will the inheritance of white demand that it is going to be dwarf also, or it can be white on a tall plant or a short plant, and vice versa, okay, can that happen or not? So, do they influence one phenotype, the genetic information for one phenotype, does it influence the transmission pattern of the genetic information for another phenotype? So, that is what is this second experiment, dihybrid cross. Let us assume it will influence, like the tall and purple will always go together, then it will boil down to 3:1 ratio, it will be like a monohybrid cross, right, and that is what is illustrated in this cartoon. Here we are taking round and yellow seeds, okay, the seed shape is one phenotype, seed color is another phenotype. So, you assume there are 2 copies for each of those 2 phenotypes, okay, it is also called trait, and you are crossing it with another one, that is, now you know how the crossing is done, remember that cartoon. So, where it is round, it is wrinkled shape and green color, and when you look at gametogenesis, then you will find one copy for each, okay, and when they fuse you will get this combination, right, it is heterozygous for both, and that is round and yellow, meaning yellow is dominant over green, round shape is dominant over wrinkled shape. And when you go to F2, if the round and yellow always were going together, then you will only get this 3:1 ratio of round yellow to wrinkled green. So, this is what you would predict had to happen. But when Mendel did the experiment, he found that not to be the case, indicating that probably they do not influence each other. So, what he found was 9:3:3:1 ratio, okay, 9:3:3:1 ratio of all possible 4 combinations. So, yellow and round did happen like one of the 2 parents, and you got green and wrinkled like the other parent, but you got the mix as well, okay, green and round, yellow and wrinkled also he got, and this Punnett square explains how that might happen. Okay, so, I am not going to go into the detail, at least that much detail I will ignore, I am sure you understand this right away. So, this immediately points out that again you need to explain this ratio, you need to assume totally no bias in which allele goes into the gamete with respect to both of them. Okay. So, a gamete that inherits the capital Y does not worry about whether it is going to have capital or small r for the other information. Okay. So, it does not worry about it, that we need to assume. And second, again in the gamete fusion during fertilization there is no such selection and it happens purely randomly. So, then only you are going to get 9:3:3:1 ratio. So, when the inheritance pattern of alleles of one gene does not influence alleles of another gene, you call that as independent assortment. Okay. When alleles of a given gene does not influence and they go separately, we call that as segregation, that is law of segregation. When alleles of one gene do not influence the inheritance pattern of alleles of the another gene, you call that as independent assortment. So, that is the second law of Mendel, clear. So, these are like the basic principles like rules of addition and subtraction in math, in arithmetic. So, that is they are the only rules, the rest of it is derived from that. Similarly, entire set of genetics is derived from these 2 rules. These are like equivalent to the addition subtraction rules in maths. If you know this, that is it, you know genetics now, everything else is derivative of this. So, that is why it is worth taking lot of time to completely comprehend this. So, whatever I already highlighted is shown here. Okay. So, the randomness, it is totally random, there are no biases. Okay. So, now we are going to consider situation that appear as if they do not follow Mendel's laws, and we will try to understand how Mendel's laws are followed there. One situation is what we call as incomplete dominant. Okay. The F1 generation phenotype does not resemble the phenotypes of either one of the 2 parents, rather it is like an intermediate. Okay. And when you go to F2, the phenotype accurately reflects the genotype, 1:2:1. To explain this, all we need to do is we need to get to biochemistry a little bit, then it is easy to understand. Okay. So, let us assume a particular enzyme, okay, that was catalyzing the reaction to make the pigment to make red color. Okay. When you had double the dose of it, remember we have already learned dosage compensation, right, and when you have double the dose and it makes enough pigment to make the flower red, but only if you have only output from one copy, that means half the quantity of the enzyme. Now, the pigment produced is, let us say, only half of what would be made otherwise, and if that is visible, you know, the diluted state is readily recognizable, then you will have this incomplete dominance. But Mendel's laws are strictly followed with respect to the genotype here, only in the phenotype you see a variation, and this is called incomplete dominance, the way the phenotype is exhibited by the underlying genotype. Only there is a difference. And so this is in carnation, so that is snapdragon, you know, Darwin's favorite plant. Then this is coronation, having very similar thing, and then there is an opposite situation where both the alleles show their presence. Okay, that you can't understand looking at flower color. A white flower cannot simultaneously be a red flower, but there are situations, for example, cell surface markers. If you take, you can have two markers at the same time, right? Let us say there are 100 receptors on the surface of a cell. Now 50 receptors could come from one allele and 50 receptors could come from the other allele, both are there on the same cell, and if one receptor bound one particular variant of a ligand and another one bound another variant of the ligand, now this cell will bind both of them, right. So, in such a situation both can show their presence and we call that as codominance. So, blood group antigen reaction is one such thing. Okay, so that is shown in this. So, I am not going to go into the detail because you will readily understand this. So, for example, you take this, you take the genotype, when you have blood group A and like the RBC surface has that particular glycosylation. So, you call capital A, and that could be both, it may be homozygous or it may be one has it and the other one does not have it. And similarly for the B, this has the B variant, a different sugar attached than what is A, and that could be homozygous or heterozygous, right. And this is also possible, as I just explained, when you have multiple receptors, some could come from one allele, some could come from the other, and both may not be present. You probably lack both the modifications and that could lack one sugar basically, and you will not have either one of the two. Okay. So, now the way it reacts with the corresponding antibodies, that is where you find that it is codominant. It show like when it is A, B, both show up. So, they have antigen for both and therefore, they lack anti-serum for both, therefore, you do not get any reaction. So, that is codominance. You understood how codominance is possible. So, in both cases we are getting to biochemistry to explain this, right. So, this is the actual blood test, so we will ignore that. We are going to consider multiple genes at the same time, so that will be in the next class.